it's just past 11. Hello, everyone. Thank you for joining us. My name is Yvonne Valancourt. I'm the director of the Nantucket Field Station, which you can see some of the grounds behind us. We are a facility of UMass Boston. We're managed by the School for the Environment, but we are open to support courses and researchers from across all departments as well as beyond the university. We sit on over 100 acres of beautiful marsh, harbor front, as well as meadow and scrub oak laden conservation land, all owned by the Nantucket Conservation Foundation. And it is my distinct pleasure today to introduce to you Nichola Hill. She is an assistant professor at UMass Boston, and she is joining us today to talk about bird flu. And Nichola is a ecologist, virologist, and a birder, and she has held some rather impressive postdocs starting in 2008. She joined us last year in 2021 at UMass. She came from Tufts, from the Department of Infectious Disease and Global Health. Prior to that, she was at MIT and their Department of Biological Engineering. And before that, she was the surveillance team coordinator for the United Nations Food and Agricultural Organization in Rome. And before that, she was out in San Francisco Bay at the Estuary Field Station with a postdoc position for the U.S. Geological Society, as well as the Department of Veterinary Medicine at the University of California at Davis. And so she has a wealth of experience and information, and I won't hold up the talk any longer. Please welcome Nichola Hill. Thanks, Nichola.
Thanks, Yvonne. I appreciate that. Very, very generous introduction. So I'll switch to sharing my slides. Let's see. Okay. Can everyone see that? Let me, oops. All right. Can everyone see a giant slide that said birds and bird flu in a changing world?
Yes.
Yes, yeah, thank you. Thanks, Yvonne. You'll have to be my conduit here. So thanks for the warm welcome. I really appreciate it. It's a great opportunity to connect with people who have a mutual love of the natural world and discuss exactly what's going on in terms of infectious disease spread. So I've been studying flu for a while now, and it's become increasingly relevant. And we'll talk a little more about the highly pathogenic avian influenza outbreaks that are occurring in North America. But just to start, a little bit about me. I'm a disease ecologist, and my two favorite organisms to study are birds and viruses. These are critters that really know how to move and spread, and it's always a challenge and a pleasure to try and study these organisms in the wild and in natural field settings like this. And this was a photo taken in Bangladesh when I was working with the United Nations there. And so in the name of studying birds and their viruses, I've had the privilege of traveling to some really faraway places. This is we're doing a blood draw from a ruddy shelduck here in Bangladesh to screen for influenza antibodies. This is a common teal wearing a satellite transmitter that we marked in Egypt. This is down here in Mongolia, where we were setting up mist nets to capture and sample wild birds from the wetlands there. And then during your downtime, there's a lot of precious opportunities to get out there and see birds. So it's really just been an amazing experience to be a disease ecologist. And so birds really are these amazing indicators of global health. They can migrate over really vast geographic distances, and they serve as a reminder of just how interconnected our planet is. So these are northern pintail that breed in Alaska, and they've migrated all the way from East Asia. And these birds are really considered quite a high-risk species for the movement of highly pathogenic avian influenza. So a disease outbreak, a contamination event, or oil spills that happen, in a far off place like East Asia might impact locations that are very, very far away, but they have a global footprint because birds and their ability to migrate over long distances ensure that the footprint for disease and these types of events tend to be very, very large. So the geographic and spatial scale that we're working at is always really, really huge when we're talking about disease ecology like this. And so there are an increasing number of pressures on bird health. So the first one is habitat loss that we all know, that conversion of natural habitat due to basically human population growth. Also, agriculture and food production is really relevant when we're talking about disease ecology too. And even that the recent mysterious disease that was causing songbird die-offs in the Midwest and made its way slowly to the Northeast was probably linked to pesticides that we use to protect crops against that cicada X predation event. And lastly, climate change. So we live in this world where birds are under pressure, and much of their migration and their annual cycle is really tied to these seasonal cues that are shifting. And so we now hear about things such as the advancing spring migration, where birds are really challenged to keep up with the green wave moving north during the spring, and then that frost wave moving south during the fall to the winter. Those signatures are getting really, really messed up, is one way to put it. And the health and susceptibility of birds is really taking a toll as a result of that. And I think humans like to comfort themselves that these types of events, environmental disasters or disease, tend to stop short at national borders. But in the reality, the reality in the non-human world is really hugely different. Birds do not care about country or political boundaries, and they take advantage. And this is some satellite telemetry tracks of a glaucous-winged gull that was collected from some collaborators in Alaska, and it shows that these gulls were able to complete, taking advantage of a tailwind, they were able to complete a total round trip migration between the two countries, Alaska and Russia, within 48 hours. They could do it literally in a breeze. It was so easy. And I think we forget how birds connect these regions across really large geographic scales and political borders as well. So we have seen a recent paradigm shift to sort of really start to think of um, animal, human, and environmental health is really connected. And so you might start to hear a little bit more about One Health. And I think COVID probably is a really good example of this, where a spillover event that happens in a live animal market somewhere in Wuhan in China might have such a huge, profound impact on the rest of the globe. But we know that it wasn't just that simple. There were many, many layers to that event, actually. And so this idea that's pretty actually de facto in a lot of indigenous cultures, where everything is connected. Western societies are pretty much playing catch up with this. And we hope that we can get there in time. And in terms of sort of making sure that we have a sustainable planet. And so I'm going to come back to this question of why is bird flu so successful? And this is one that I spent a lot of time thinking about. And we know that this tends to be a virus. Well, now is the time really where we're talking about viruses that originate from animal reservoirs spilling over and just spreading rapidly and on a global scale. COVID-19 is one example, and now avian influenza in wild birds and poultry is starting to surface again as a real threat. And so why is it that viruses that originate from animals like this, including bird flu, tend to be so successful? And so one of the theories that I'll be sharing is that they have a host that flies really, really, really far. And so we've been discussing that in terms of water birds, which are considered one of the primary and original reservoirs for avian influenza. They move, they just cover huge, huge distances. And the success of the virus is really largely resulting from the fact that these are birds that migrate over large distances. And this migration links populations that are infected and susceptible, and there's lots of turnover of naive individuals that can become infected, and it really facilitates spread of the virus. And so I'm sure that a lot of this group has probably checked out eBird, where you can just watch and just look at their data visuals for days, and just be completely wrapped up in the way that wild birds move. And so this is just an example of a blue-winged teal population wide, and how they really do connect parts of, I can play it again, parts of Alaska, and then all the way down to Central America and Guatemala. It's just astonishing to see how the vast distances that they actually do cover. So it's not only the fact that water birds, which are the primary reservoir for avian influenza, that they fly, it's also because they swim. So we're not talking about land birds or songbirds or passerines. We're talking about birds that when they move, they actually are attracted to water bodies when they stop to recharge and feed, or breed, during different parts of their annual cycle. And the fact that they both fly and swim, this is really good news for the virus. When the RNA viruses don't last very long outside of the host. And so the environment really matters. It can really dictate whether the virus survives for a short period or a very long period. And when this virus is shed into the water, usually via fecals, just droppings, that the birds excrete while they forage around in wetlands, the virus is able to be preserved. And it also circulates, and it creates the ability for the virus to actually move between hosts that are susceptible. So, this is a really happy place for the virus. There's a lot of studies that show, and we've seen COVID-19 is a really great example how we're able to recover that from wastewater and sewage. When viruses are outside of their host, they really like to be in cold water. It really helps their survival. So the wild birds, water birds specifically, have this really unique ecological niche that allows the virus to be quite successful and to thrive. And so there are exactly 112 different subtypes indicated by these green boxes that have been reported from wild birds to date. That's a huge amount of viral diversity. And you can see the two axes here are the hemagglutinin on the top, and on the left are the neuraminidase proteins that are actually on the surface of the virus. Don't worry too much about that. What matters really is, well, a good example is the difference between two of these green boxes is orders of magnitude higher than, say, the difference between Delta and Omicron for COVID-19. So if that just gives an impression of just how large the viral and genetic diversity is that wild birds host. And so when a bird's infected, it's probably equivalent to a human getting a mild case of, a cold or something like that, where you feel a bit sluggish, you don't really want to get out of bed, maybe you call in a work from home day, and then the next day you're fine and you're out the door. And it's probably the equivalent, to the point where we actually can't tell by looking at birds whether they're infected or not with these low pathogenic forms of the virus. It's actually impossible for us to tell. We know that there's probably some really mild symptoms, but humans generally are pretty bad at actually detecting this. And so it's not easy to tell just by looking at a bird whether it's positive or negative when we're talking about low pathogenic forms of the virus in wild birds. So the other reason why bird flu is really so successful is because humans like chicken. And the honest truth is that a lot of the globe thinks that birds are delicious to eat. And so that matters because while we know that wild birds are really at the center of the epidemiology of bird flu, the virus infects many other hosts, actually, including marine mammals, which is an active area of research for this group, coordinating with the Tufts Lab under Dr. Wendy Perrier as well is a really interesting area of research for us. We know that it also spills over into cats and dogs and domestic animals. There's endemic circulation of influenza viruses that occur in bats as well. We've found new strains of the virus in completely distinct subtypes that we didn't know occurred. Horses as well, and of course humans. But transmission of the virus from wild birds into poultry is actually the most common transmission route. And in part, that's because poultry is one of the cheapest and most abundant forms of protein in both industrial and non-industrialized nations alike. And we also know that humans quite like swine or pork as well. So these animals that they account for a lot of sort of head of agriculture worldwide. These tend to be the pathways that can be quite common for spillover events to occur. And so we know that a lot of our food sources are basically from industrial agriculture. And the mantra there is live fast, die young. So these are really genetically very, very similar birds. And they create an amazing sort of susceptible population for the virus. When it spills over from wild birds, it can mutate into highly pathogenic forms in this type of host because they pose absolutely no resistance to it. And so the virus can actually become quite virulent and not experience any negative feedback from it because there's constant turnover of new birds being introduced into the population. And so that transmission between wild to domestic is exactly the route for which highly pathogenic forms of the virus occur. So going back to this giant plot where we have our 112 subtypes that are low pathogenic in wild birds, you wouldn't know it by looking at them, doesn't seem to have any clinical signs that humans can detect. These three subtypes that are shown in red are actually, these are human seasonal influenza, which again originate from wild birds, but have to move through an agricultural host before they actually became endemic seasonal strains. And this is part of the reason why we get an annual influenza shot, which usually includes, 3 of these subtypes and sometimes a quadrivalent vaccine if it includes influenza B as well. And then these additional 6 subtypes shown in yellow, so these are the ones that we're really, really concerned about their potential to move from the wild bird reservoir and spill over into animals, including humans. And so H5N1 is there, and this is on the WHO's pandemic watch list. They're really, really concerned about these particular subtypes of the virus spilling over from wild birds and actually becoming pandemic in humans. But it's not, there always has to be an agricultural host in the middle, and I want to really, really sort of reinforce that. Wildlife to human spillover of this virus hasn't actually ever been documented. There needs to be sandwiched in the middle of the wild animal and the human is this agricultural host, which is responsible for this generation of these highly pathogenic forms of the virus. They're really good at adapting, amplifying really big explosive outbreaks, and then we tend to see highly pathogenic forms of the virus periodically spill over into humans. A very, very, very rare event, but when they do, it can be actually quite lethal, averaging 50 to 60% mortality rate in humans. And so we're always watching for these interfaces between wild and agricultural, first off, to see if we can always get ahead of the virus. Usually by the time it's in agriculture, it's a little bit, the consequences can be a little bit more high stakes. And so the goal, a lot of what we do in disease ecology in my lab is we're looking upstream at routes for transmission between wildlife and agriculture as they're occurring. And so I wanted to show you, this is a video of when we're in Mongolia early on, this was an H5N1 highly pathogenic avian influenza outbreak that was impacting bar-headed geese. And this was a population that we were able to put transmitters on, and they moved incredibly far distances. But this was one of the birds from that population that we didn't manage to mark, and it should reveal some of the clinical signs of highly pathogenic avian influenza. And you probably heard one of my colleagues speaking in Mongolia really tensely about what was happening in the background there. And, this is disturbing to witness. You're seeing a lot of the neurologic symptoms, the walking around in circles, the lack of balance. And so this is no longer low pathogenic. We can absolutely tell that this is an infected bird. And geese were really prominent. They played a really big role in the movement of highly pathogenic H5N1 during some of the original outbreaks that were occurring throughout Asia during some of the earlier phase of the virus as it circulated. And so the third reason why bird flu is super successful is that viruses are really simple and they're really sloppy. And what I mean by that is that they're sloppy at replicating. And so we know that viruses are really, really, really bad at copying exact copies of their own genome during the replication process. So, for example, here are our RNA viruses like COVID and avian influenza. And so they're pretty high on this scale of the mutation rate. And down here are all of the cellular organisms like humans and bacteria. And these tend to have much, much lower mutation rates. And so it's actually thought that there tends to be this negative association between mutation rate is often really, really small organisms and higher eukaryotes that they're during replication of the genome, there doesn't tend to be as many mistakes or mutations or errors that are made. And that leads to larger genome sizes and more complexity in that direction. So viruses have sort of done well in this niche of just being really, really small, really, really simple, but really, really bad at making exact copies of their own genome. But what that does is it means that there's a huge number of individual copies of the virus after completion of each of the replication cycles that creates a number of variants that has the potential to actually become sort of fine-tune their genome and then adapt to a particular environment. So this is sort of why viruses, part of the reason why viruses are thought to be one of the most successful organisms on the planet. And so that the fact that there's so many mutations that accumulate with every replication cycle means that they're able to adapt really rapidly, and they're constantly fine-tuning their genome. And this is a really good example. So this is actually COVID-19 data, which probably a lot of people are starting to be familiar with. But this is a period that just coincides with effectively 2 years since the start of the pandemic, and how the evolutionary pathways are really expansive. Like it just there's a lot of genetic variation in starting with the original source of the virus until now, and we're just playing catch up with how many variants occur. And so again, this is a really good reason why the virus mutates quickly. It's able to adapt, and we know that with selection you need to have a lot of different mutations occurring for adaptation to occur in a population and for an organism to survive. And so viruses are sort of the pinnacle of evolution in that way. And so, really, the next question is, why did bird flu spread to North America? So, I showed you some footage from Mongolia from about 2009, and it's been circulating in parts of Asia, East Asia, Central Asia, and also Europe for since about 1996, when highly pathogenic avian influenza first burst onto the scene in a domestic goose in Guangdong province in China. And so it sort of moved in fits and bursts throughout Eurasia for that period. And then we started to notice this big change in the virus's geographic footprint. And it started to move more extensively into Africa, for instance. It's now completely endemic in Europe and the Middle East. Uh, it has a much bigger, it's also endemic in parts of India and Bangladesh, in Eastern Asia as well. And we also see it periodically in Siberia as well. And so we know that the geographic footprint of the virus is definitely expanding. There's this big distinct change in the epidemiology of the virus at that period. And so, in that second phase of evolution of the virus, we did see that first introduction into North America in about 2014. And so that was really the first time that it had moved. And there's fairly compelling evidence that it moved, it circulated, it was entrenched in poultry, and then there was a migration event involving wild birds that moved north during that spring migration. Huge breeding sites in Siberia where lots of different migratory flyways overlap. And then the virus moved south through the Pacific Northwest. And it basically at that point just really radiated across the US at that time. And so we know in 2014, it landed here in late 2014, followed by a period of outbreaks in 2015. It really savagely, yeah, it blew up in the poultry industry, particularly in the Midwest, and it spread to 21 states in the United States. And so this event actually prompted the culling of about 40 million chickens and turkeys. And there were quite serious um, consequences of this involving labor shortages, and the price of eggs went up. And for anyone who was watching, it was a pretty harrowing event to bear witness to. But then it ended. And the US has been considered, and North America at large has been considered free of highly pathogenic H5 since about the mid to late 2015, since that period. And so we've had a long stretch where we haven't actually seen it, and people thought, okay, we're done. This event, we've been able to sort of wrap this up. It circulates endemically in Eurasia, but these events where they spill over and become introduced into North America are really rare. So this was our group who actually took all of these viral sequences that are available, and we looked at them, and we tried to basically determine which wild bird and domestic bird taxa are really responsible for this fast virus dispersal rate. And so these are all of the highly pathogenic H5 genomes that we could collect. And we categorized them according to these different bird taxa. And what we clearly noticed was that when the virus moves into wild birds, it can really, really move quickly, far more quickly than it can in domestic birds. And so, what was also really interesting, and it came out as a bit of a kind of standout result, was that gulls, distinct from ducks, were really, really responsible for very rapid spread of the virus. And so we know that domestic ducks and geese and poultry play a really important role for amplifying an endemic, continued ongoing circulation of the virus. But when it does move over into wild birds, they can really, as we know, move it really rapidly, really quickly. Um, And you can see land birds here are a bit of an interesting comparison as well, where they just don't tend to move it as fast as water birds. And also, this research was interesting where we looked at the geographic footprint of the virus just using viral genomes. And of course, this virus is endemic throughout Africa now, Europe, throughout Asia as well. And we had this one introduction event that we think happened via the Pacific flyways in 2014 and moved into the Midwest poultry belt. Um, and it didn't seem like there was any evidence of a sort of transatlantic movement. But when you compare this subtype to low pathogenic subtypes, for example, H13 and H16 that circulate widely in gulls, but these are low pathogenic, what we were observing was that interhemispheric transmission of these viruses in wild birds moves no sweat across the Atlantic coast from Northern Europe into North America. And so there was absolutely a precedent here for this to take place. And we knew this comparison of high path and low path was revealing maybe this is only a matter of time. We know that wild birds are completely capable of moving this virus over long distances. We're just waiting for a point where the virus starts to adapt enough to wild birds where it's not pathogenic, it finds this sweet spot and it can be carried long distances by wild birds. And we think that that is the big event that happened that was distinct from 2010 onwards. We have a new lineage of the virus, which is much lower virulence in wild birds. It's starting to approach becoming low pathogenic. It's starting to look and feel like a low pathogenic virus, depending on the species. And so we do feel as though many events, the virus's evolution, the increasing connectivity of wild birds along the high Arctic, particularly with climate change. There's a lot of, residency, but birds are not moving the same that they used to through these areas. There's not the same strong seasonal cues. Really, this perfect storm of events that meant that we saw that introduction event into North America via the Atlantic in late 2021. And so this is some work that's being performed by a graduate student in our lab, John Gass, and this is specifically just where the birds were moving. Imagine this is the high Arctic, so we've changed the projection of the globe now, so it's sort of polar centric. And what you're looking at is mainland Europe over here, connection via Iceland to Greenland, and then North America on this side of the chart. And it's absolutely clear that gulls do an impressive job of really connecting those parts of the globe, along with ducks and geese as well, and shorebirds too, coming down also sort of from the Arctic tundra. So a good mix of species are really well connected across this area. But there's no doubt that gulls were sort of the smoking gun, really, in this scenario where the first detection of highly pathogenic avian influenza in North America was detected in late 2021 in Canada, and these were occurring in great blackback gulls. And so it seemed to be, and a lot of people had suggested, and we'd looked at the data, that if it was going to move via the Atlantic, it was probably going to be a bird that could really move long distances like a shorebird or like a gull, whereas the Pacific introduction was specifically waterfowl. And so there are some really distinct differences between the roles that different species play in the epidemiology of the virus. And since that time, so this was in about late 2021, we detected the great blackback that was positive, and then since then it's sort of uh, been amplified and moved largely by dabbling ducks in the US. And so the first detection occurred in January in American widgeon in South Carolina. And we've also noticed since subsequent detections in mallards, in New Hampshire, in northern shoveler, in widgeons, a lot of the dabbling ducks are at play. But I will also say that this is hunting season, and a lot of these detections are just opportunistic. So we're sort of focusing our efforts on wherever we can get hands on birds, and very easily, because of the hunting culture in the US, dabbling ducks and hunting go together. And so it probably isn't a huge surprise that we were going to inevitably detect it in dabbling ducks in sort of the South, and places where hunting culture is more common. And so there's huge, huge surveillance gaps, as we know. Um, and so the roles that other species play in how the viruses move to date are completely wide open. We have nothing other than dabbling ducks to show for how the virus has moved in the US. And we know that we have a lot of work to do in terms of figuring out all of those various pieces of the puzzle, and fitting it all together to figure out where it will go next, and how widely it will spread. And so, in conclusion, just to wrap up this section of the talk, I wanted to sort of try and end on a bit of a high note in terms of how we approach controlling bird flu. In the short term, when it gets into agricultural populations, uh, so culling and depopulation of farmed birds in the infection zone. So they usually draw a ring. Um, it's basically called ring culling, where you say, a 5 kilometer radius around a particular farm that is found to be positive. There is no animals that move in or out of that farm, and everything in that zone is depopulated. Um, of course, disinfection of farming and live bird markets is really, really common in parts of East Asia. We do have live bird markets as well in North America. They're not as common, but they're also an important part of this as well. And what we call all in, all out practices for the turnover of poultry. So we make sure that birds are really quarantined and flocks are either depopulated, there's no single animals that move in or out while a farm is under surveillance. It's really, really important. And so that's largely where we hand off to the USDA to help. There are vaccines in development for poultry. It's not the preferred measure for control of the virus. And so there's a lot, often because it requires multiple doses in succession. And so it hasn't been, it's really the rollout of vaccines in places like, the Middle East and East Asia, where it's more difficult. The turnover of poultry means it's quite difficult to actually get to to make sure that poultry flocks are vaccinated adequately. And then in the long term, sustainable poultry agriculture, we kind of need to consider the role that industrial agriculture plays in making our wild animals actually more susceptible to disease spillover. And of course, conservation of wetlands and wild bird habitat, making sure that there are places for animals to basically be wild, and not to have to constantly have an interface with humans and domestic animals as well. And so that preservation of non-human spaces is incredibly important when we're talking about trying to stop diseases that have the potential to be zoonotic and spillover. But also, I like to think of it's what you eat as well, to a large degree. And if we are reliant with a huge global population on industrial agriculture, it has consequences, absolutely. And so I'll end this part of the talk with what can we do? What can you do as community members who care about nature and the fate and the health of wildlife and wild animals and wild spaces? So there really is this huge role for community scientists and the public. This response to the influenza outbreaks at the moment has taken us by surprise, as you can tell. So the detections are largely happening from hunter-harvested animals, and there are so many gaps in our understanding. And so I think a lot of those activities like beachcombing and understanding what normal looks like in your own backyard are just so, so important. And I will say that as someone who studies flu, I probably get to the field a couple of times a year, if that. I love it. It informs so much of how I approach science, and I love the process of observing and listening to these natural systems. But I'm not out there enough. And I do think that because I'm spending time in a lab or looking at viral genomes, and it's a difficult balancing act for those of us. And I do believe that the community are very much like the eyes and ears of what's going on out there in nature, and figuring out what is baseline, and then when is something unusual? When are we seeing incidents of unusual activity in wild animals and birds? And I think that those types of things are really, really useful. Um, Keeping open channels of communication. And I know that Yvonne is spectacular at this, but talking to people, trading information, making sure that we're informed about the situation. And social media has a huge role to play in that, about rapid dissemination of information as it's developing. The situation with bird flu at the moment is really, really fluid, and we don't quite know what the virus looks like or if it's hit the Cape. These are all open questions that our labs are actually trying to establish and get at. And lastly, ideal at this stage not to handle dead birds, but to really report them instead. And so, as I was showing in some of those charts, we've never had a virus from a wild bird move over directly into humans. So it's really considered a low risk event for that to happen. The virus typically needs to adapt and mutate in poultry or swine before it spills over into humans. But regardless, it's about taking universal precautions. Um, the other side of that is that we don't want birds left that might be infected to encourage scavenging, and it becomes a potential transmission route for raptors and other species that interact with dead birds, even marine mammals, right? Or foxes or coyotes. So this is definitely an issue. So if we do see dead birds, it's about not trying to handle them directly, but reporting them instead. And if they are handled, it's about taking the proper precautions and making sure that we're gloved and masked up, and the animals are double bagged before anybody is handling them. So we can talk more about that, about what the infrastructure is like on Nantucket and how to respond more effectively to the outbreak. And with that, I'm really happy to take any questions. Thank you.
Well, thank you so much, Nichola. That was really excellent and interesting. We do have a variety of questions that have come in. From let's see, Val Hall sent in a couple. Can you see them? I can read them to you, but
Yeah.
I think the first and overarching one was, well, what can we do to alleviate this problem? That came from Val, and she did seem to think you could answer it, but I do think that particularly where we are on Nantucket, we have about half of our island conservation open space, lots of birds migrating through, and lots of people working in the realm of conservation and wildlife management as stewards. How do we approach this from your perspective? What kind of recommendations can you give us in respect to what can we do? And then I think there's a slew of comments and questions about what do we do if we, or what should we look for symptoms, and then how do we best address an outbreak were we to see something?
Yeah, I think these are all great questions that we could chew on for days. I think this is the starting point. And Yvonne asked me to give this seminar, and I thought, "Oh, I don't have time." And then I was like, yes, I do. Like, there probably isn't anything more important than starting to open up the channels of communication, and talk about bird flu and what people are seeing and hearing about it. And so understanding that surveillance is really incredibly patchy. And so, and I think Massachusetts and New England is under the USDA's jurisdiction, but they're often not available, and there's just limited resources to respond all the time to what people are seeing. And so, sort of, this is kind of like the moment where we can start to talk and develop resources and infrastructure for responding, I think. And it sounds like there's already a lot going on Nantucket, in terms of like a community that is constantly in contact and trading and sharing information. And so I think it's about tapping into anytime, people are going out into nature to observe, with a pair of binoculars, and sort of saying, is this, continuing to report even just baseline normal activity to eBird is really, really important information. So we get an idea of what baseline is, and then what's unusual, or a kind of different activity that we're not used to. And so, in terms of handling dead birds, there isn't a lot of information or guidance on that right now. I think it's basically a case of, and knowing that dead birds are actually not atypical, right? So it can just be like a really big storm event, and you get like a bunch of seabirds that might be a little bit malnourished and, under condition, and so they wash up, on the shores of Nantucket. And so there are some sort of baseline activities that we know that that's not related to the virus, but this is the moment where we're saying, if we see like large groups of birds that are, or anything above baseline, but also bearing in mind with the charts that I was showing you, and you also you now know what a neurologic goose looks like, which, walking in circles, lack of balance, but also worth saying that the pathology of this virus is now really wide ranging. And so the virus has done an amazing job of evolving to, this new reservoir. And so there are birds where we don't know that they're wild birds that we don't know that they're infected, and they're just normal behaviors, but they're actually shedding virus. So it's the whole spectrum of symptoms that we're seeing for wild birds, which makes things incredibly difficult. Um, but yes, this is the starting point, really. And I can certainly work on. I'm getting more information as we go from the USDA. I was saying to Yvonne, we're just in the middle of trying to figure out our response. I'm setting up my lab at UMass, but working in collaboration with my Tufts lab, where I was a former member, that have a surveillance operation. And we do a lot of diagnostic testing in that lab, and figuring out, what's the appropriate response when we come across wild animals and not just wild birds, because we know that it's spilling over into mammals, working its way through food webs. So there's a number of European red foxes that have now been reported, with highly pathogenic forms of the virus. We know that marine mammals can get it. So gray seals, I don't know if Wendy's on the line, but gray seals also are infected as well, with highly pathogenic forms of bird flu. So we know that it has this ability to spill over increasingly. And so it's sort of a bit of a fluid situation where we're trying to figure out what to do.
Wendy is on the line. We also have a few other questions and comments. Certainly, we have, as Neil Foley points out, we have a lot of birds and a lot of mortality, and Danielle Odell also pointed this out. How do we know what baseline is if we don't monitor the death counts that we see? And of course, as you pointed out here, we get mortality events just from storms. And
Yeah.
so if we were to test dead birds, I mean, you're saying stay away from them. So it's something we would only want trained people
I
to
I
swallow.
that's exactly the gray area that I need to get back to. I don't want to be seen. It's slightly an optics issue here, but I don't want to be encouraging people to get hands-on with wild birds. I would say at a minimum that people need to be, masked up and using gloves, which are actually fairly common supplies that you probably have from COVID. So I think that that's useful. And then, I think it's possible to bag birds as well, like to double bag birds and then take them to the local animal hospital and report birds that are sick or injured, absolutely to rehab facilities that are available on Nantucket. So I think those should be really open channels of communication right now. And I
Um,
know that you're doing that too.
so something that's come up as we discuss, how do we do that here on the island? How do we best prepare ourselves if we were to have an outbreak? The question repeatedly comes up, is there a field test, a quick, rapid test
Mm,
for bird flu, or is there one being
great
produced?
question.
That would be a
Great
really
question.
handy thing to have. What we're concerned about is people here tend to walk outside a lot in our lovely open spaces. They see what's a ill bird or something in need of assistance, and they capture it and
Yeah,
they bring it to the hospital, or they report it. And
right.
will that, if the hospital then starts to say, sorry, we can't take it because of bird flu outbreak, it'd be really nice to know if the bird in need of assistance is a carrier or is ill with it. And so
Yeah.
if there's a rapid tester or someone who wants to engineer one.
They're not in wide circulation, I will say that, and it's a bit of a function of the virus evolving so quickly. And so they do have rapid tests for H5, so older lineages that were circulating in Eurasia in poultry. Whether those rapid tests, they're sort of like these dipstick tests, are still useful against this lineage of the virus that seems to be successful in wild birds is not clear. And so still, and we sort of know from COVID as well that a PCR test is really the gold standard. And I can say that the Tufts lab um, is, this is a real active area of research for the Tufts Lab right now. And so we are doing our best to keep up. We're constantly having conversations with people in the wildlife rehabilitation world about what animals they're seeing, whether they're sick or symptomatic, and, trying to get samples into our pipeline. The parts where collection by the public is basically, I will need to get back to you. That's not clear cut at the moment. Mostly we say that this is very, 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 very low risk of it spilling over to humans, but I can't recommend in my current professional capacity, people going and getting hands-on with birds that are symptomatic or ill right now. So leave that with me and I can see if I can do more digging.
This has been such a great thing for you to do for us. Thank you. I don't know if there's any other lingering questions or comments. We have developed a working group down here to inform ourselves and share information. I am looking at creating a citizen science observational platform somehow. And would you know, so that we could develop a baseline in general,
Right.
but also so that we might begin to recognize when we do have mortality, or if you're testing to have data match up with what we see. So I know on eBird you can write all sorts of details, but do they have a way to, do you know if that is filterable?
Links to disease?
No, on eBird, so can you filter the mortality events?
Oh, I'm not sure. That's actually a good one to look into further. Um, I think it's potentially. So this is a really interesting area that I think is a bit of a gap, really, is scavenged, sorry, or is basically dead birds, and how to handle them that are just seen out in the open. And so there is great community science groups on the Pacific Coast. I mean, you've probably heard of them, COAST, COASST, and they have excellent protocols. Yeah, I can share a link. They're absolutely amazing, and they are focused on how to basically do beach combing that produces scientific data. And so people record the time and the pace of which they were walking, basically doing a transect, what kind of debris they observed, whether there was plastic, what were the weather conditions, whether they saw anything unusual on the beach when they were doing that. And so there's tons and tons of potential, I think, to get maybe like a small Nantucket version of that going, especially during that period. I think there was some groups. So there was Dr. Julie Ellis from Tufts, and Dr. Sarah Cauchane, I think, as well, were instrumental in starting to develop an Atlantic coast version of that program to do. But it takes like a lot of community buy-in and input to get it going. So I think now would be a really good time to revisit how to do such a program.
What a great idea.
Yeah,
Yeah.
and what I will also say that I think Wendy can comment on with the wildlife rehab stuff. Yes, we're in a situation where wildlife rehabilitation groups are getting shut down and quarantined, so it's an all in all out policy where they can't take in any more animals because they have a positive bird that is in their care. And so we're starting to see that where rehabilitation centers and animal hospitals will be under the jurisdiction of the state veterinarian, who will say, this, we can't allow any more animals in or out at this time until this positive animal has been cleared. So sort of expect for that to become more the norm and for animal hospitals to get a little bit maxed out. Wendy, what are your thoughts on that?
Yeah, I think you hit all of it right on the head. So there's an avian facility in Delaware, as Nichola was just mentioning, that has closed down bringing in any new animals until they're able to take care of the situation that is currently going on, and it's unknown exactly when that will be lifted. The bottom line is really that it's just a very rapidly developing situation, and each state is working on figuring out how exactly they're best going to respond to the situation. So it'll be really important that people are working closely with the various agencies that are available to them and keeping tabs on things as they unfold, almost hourly, definitely daily.
Yes. Yeah. Yeah, 100%. And I think the Tufts lab might be one of the only ones, I think, in Massachusetts that are actually accepting samples and doing diagnostics for bird flu right now. So as far as I'm aware, it's like the diagnostic capacity is basically us until it gets escalated to a federal or state level. So yeah, we're trying to be like the back end operation for a lot of the animal hospitals and facilities that are taking in birds and wild animals at the moment.
That was a large question we had. So if we're reporting, exactly who should we be reporting to? We do have an environmental policeman down here. We don't want to flood him with information, but we want to report appropriately to the best source. And so we've been digging up information online and have found the operational manual, but it is from a few years ago when the outbreak occurred, and right. So there's information there. But would do we just contact U.S. Fish and Wildlife or Mass Agricultural Resources?
Yes, I think to the best of my knowledge, it is basically all of the decisions are being made at the state level. So the USDA and the US Geological Survey are handing off to the state until it becomes like a national emergency, from what I understand. And we're not quite at that tipping point yet, where the feds come in and take over. But basically working up basically the reporting chain of command and this is sort of where I'm learning as well, being new to UMass, is what that the chain of command looks like on Nantucket and locally. But I think that would be like a really good task to figure out exactly how that reporting chain should go, so that local resources aren't completely overwhelmed. Yeah.
Well, thank you again, and thanks everyone for tuning in. There's lots of comments and thank yous, and this was very interesting
good.
and informative. So it's not only myself, but many people. Thank you, and thanks, Wendy, for joining us. Everybody look forward to a talk by Wendy. We're coming up with the date, but this will be a continuum of this topic and other similar things as Wendy will share with us. But thank you, Nichola, so much. And anytime you'd like to join our avian influenza work group, or we call it HPAI Watch on ACT or something like that, would love to have you
Sure.
check out what we're up to. And
Yeah.
we're trying to coordinate swabbing and sending samples directly to Wendy.
Yeah.
But thank you again. And unless anyone else has a question or comment, I can give you your time back. Let's see. Yeah, I think that's it for questions. Thanks again.
Sure. Happy to answer any more questions that might pop up. There's some good ones. A couple here about, there was a good one about rapid tests, yes, and backyard poultry, yes, definitely with the Pacific flyway introduction of the virus in 2014. After it moved from, it was first detected in wild birds, it was then detected in backyard poultry. And so that can sort of be a loose sort of well, it can be an interface, obviously, between wild birds and poultry, but I also think of backyard poultry as a slight sort of dead end. Like there's no big movement of backyard poultry across the country as is done with industrial agriculture. And so they're an interesting interface, but usually, so absolutely keep your eyes on backyard chickens as well. That's going to be also really, really important. Um, but I think there's, because they're usually so few birds in a flock, and they don't move around, they're not traded. Usually, if there's an infection in chickens at that point, you'll either have animals that are, falling over with imbalance, and it is highly fatal in chickens, you will know. And then at that point, the outbreak is over. Usually it's a dead end for the virus at that point. So yes, worth keeping an eye on backyard chickens as well. Sorry for that scary last comment to add there. And there's a question about land birds that I think is really interesting as well. So yes, highly pathogenic H5 spills over into land birds. So songbirds, sparrows, corvids, crows. I mean, we've seen it in like geofalcons and even snowy owls. I mean, it's like once it works its way through, usually the food web, there are a surprising number of birds that can get infected. I would say land birds are not this species or not this taxa of bird that are going to play a big role in amplifying. They're usually, they can carry the virus, but they're basically not as good at hosting the virus as, say, waterfowl. But should you wear gloves? Should you have higher levels of PPE? Yes, if you have them from your COVID supplies, I can't see that it would hurt, but I also don't think at this stage land birds and H5 is like a big um, pressing risk for pandemic spillover of the virus. But absolutely worth keeping an eye on the health of those birds as well.
Well, unless you're getting any straight comments sent to you, I'm not seeing any more
I think
questions.
that's it, yeah.
We could keep
Yep.
talking. We could keep asking things and discussing, I'm sure. The thought of what if a large number of wild birds do become infected and there is mortality. I mean, what do you do in a situation like that with them? I mean,
For a large wild bird outbreak, so they have occurred, and they tend to. So Qinghai Lake and Poyang Lake in China, in Central Asia, were big epicenters for wild bird outbreaks during earlier versions of this virus, where it was like highly pathogenic in wild birds, and it hadn't sort of started that process of co-evolving. And they basically, there was big discussions about culling wild birds at that point, about any bird that lands on that particular, and it's this really pristine, gorgeous wetland, and there was discussion about really trying to do captures and cull birds, and usually the guidance is hands off wild birds. Like, wild birds are wild, the virus is naturally acquired. This isn't a spillover from a human, for instance, and so we need to sort of respect that boundary. And so I think it's fine to be, testing wild birds that come into our care and doing sampling and surveillance where we can actually ensure that we're being safe about it. I would say anything where there's talk of like intentionally going and doing captures and rounding up of wild birds with that in mind is absolutely off limits. But if you do see events where, and I can imagine it would be somewhere maybe like an inland lake or something where there's transmission of the virus in water, potentially. It's sort of tricky with wild birds to see that phenotype, but they may occur where there's like more than a handful of birds that are sort of have those types of symptoms, where they have neurologic symptoms, basically. What can you do? Report. Honestly, like document and report and open those channels of communication. And I think at that point there would probably be an attempt to sort of make sure that people aren't moving in and out of that area, that traffic is minimized to it, and it might, there might be a quarantine zone around it.
Last quick question. I know you have to go very soon. Seth Engelberg did bring up another question. So we spoke about rapid tests, but what about eDNA? Does anyone do testing in that?
Oh, interesting. Yeah, eDNA has its pros and cons. It's not widely used because RNA doesn't survive super long outside of the host. So it's not straightforward to do those types of approaches. I think this is an area where we know that there's like a waterborne transmission phase for the virus, and it's important, but that time frame is very, very limited. And it usually has to be met with like if there's waterborne transmission, there's usually a high density of birds, so it can move quickly into a living host, rather than have to sort of persist in the environment like that. I think we're getting better at environmental detection of viruses. It ramped up, we've got a lot to learn from COVID about applying that to animal origin viruses and what we can do in natural settings, I think, as well. But I think that's a really interesting future area of research. We do it on small scales. It is labor intensive. The concentration of it is not straightforward. Again, Wendy, do you have any additional thoughts about how we could scale up environmental sampling that we do? Oh, she may have stepped off.
I'm not sure if I'm able to unmute your, oh, it looks like I have been able to unmute. Yeah, so the environmental component is super interesting. So we work with people that are from University of Maine. Christina Kamen is looking from a genetics point of view from the host, but less so on the virus. And the virus is something that we've been trying to add as well. In part, the problem kind of comes down to how do you filter a pond? How do you filter the ocean? So it's taking this dilute source of a sample and trying to figure how you can get a concentrated sample that you're actually able to then test in the lab. And we've tried several different things. It's a work in progress. So it's not great at sensitivity yet, but it is definitely something that is worth pursuing further. And it is definitely possible to pull samples out of whether it's a sediment or, you know, a soil core or water directly, it is possible to detect influenza for sure. So that's worth further pursuit.
Yeah, it's definitely been done. Recovery of influenza from water and from sediments, you know, by members of our lab. It's again, as Wendy was saying, it's that scaling up question, which can be. We'll work on it. How about that? Leave it with us.
Thank you again. I know you have to go. We'll continue this conversation. It's really interesting, and there's so much that we could do for basic baseline surveillance down here. So I'm looking forward to digging into that with citizen science observation. So anyone listening who's interested in doing that, you should reach out to me. And thank you again, Nichola, and thank you, Wendy, for joining us as well and fielding some questions.
Definitely. Thanks for having us. It was a pleasure. I hope to get down there soon.
We hope you get down here soon too as well. Thank you.
Thanks all. Bye.
Bye.